Hello and welcome to my channel. I'm Dr. Shivika Sethi, the microbiology faculty at Marrow. So the NEET 23 exam is approaching soon, and for the last few months I had been receiving several requests for putting together microbiology images. However, this is a Herculean task because microbiology has countless images. So this is a small effort. I'm just starting. This is part one of all future videos on microbiology images. In this section, we're going to be covering all the important things that a microbiologist would see or do to identify bacteria in specimens or after isolation on culture media. Right? So basically, we are going to be seeing the stains, the microscopic morphology of bacteria, the important tests that are done to identify the bacteria in the laboratory. So our first image is gram-positive cocci. I hope you all remember gram-positive take up purple stain, gram-negatives appear pink. So these are seen in clusters. So this is definitely Staphylococcus. And all Staphylococci are gram-positive cocci, which are seen in clusters and they are Catalase test positive. This is a picture of gram-positive cocci seen in chains. So this would tell us that this is Streptococcus. So all Streptococci are gram-positive cocci seen in chains, and these are Catalase test negative. One exception to Streptococcus is Streptococcus pneumoniae, which is seen in generally pairs or sometimes short chains. Now, this is the microscopic appearance of a CSF specimen, rather CSF sediment, and of a patient with meningitis, and you can see the gram-positive cocci, some of them which have a flame-shaped or lanceolate-shaped, and these are seen in pairs or in short chains. Okay. Next image is of Neisseria. These are gram-negative diplococci. On the left-hand side, this is the CSF sediment of a patient of meningococcal meningitis. The blue arrows are pointing to the gram-negative diplococci. And on the right-hand side, this is the microscopic appearance of a urethral discharge of a patient with gonorrhea. So these are gonococci. So remember Neisseria, these are gram-negative diplococci. Do not get confused in the exam. Suppose you are shown the image of a patient with meningitis and the CSF sediment picture is shown. In your hurry, don't confuse the Neisseria from Pneumococcus. Neisseria would appear pink and Pneumococcus would appear purple. Our next image is of Corynebacterium diphtheriae. This is the gram stain picture, and you can see the gram-positive rods which are showing club-shaped swelling, and they have the arrangement, cuneiform arrangement. Cuneiform was the language of Mesopotamia, or it is a Chinese letter arrangement. Because you can see the bacteria is arranged somewhere in V's, somewhere in L's, or sometimes you can even see them arranged in parallel chains, which are called as palisades. Now, this is the Albert stain of Corynebacterium diphtheriae. This you can see the bacterium, the cytoplasm of the bacterium has taken up green color of malachite green, and the volutin granules, Babes Ernst granules, or the metachromatic granules have taken up the blue stain. So in the Albert stain, the main two dyes that we use is malachite green and toluidine blue. Okay, so green cytoplasm with purple granules, think of Albert stain. However, please don't confuse it with the gram stain. Gram stain, the bacterium would appear purple in color. So these are gram-positive cocci, rather gram-positive bacilli, which are arranged in chains, giving it a bamboo stick arrangement. So this is gram stain of Bacillus anthracis. Special stain is used to demonstrate The capsule of Bacillus anthracis, this is polychrome methylene blue, and on staining with this dye, we see the bacterium has taken up purple color stain, the cytoplasm, and there is a pinkish amorphous material around the bacterium. This is the polypeptide capsule of Bacillus anthracis, and this is called as the Mc-Fadien's reaction. The capsule taking up the pink stain, the cytoplasm taking up the purple stain of polychrome methylene blue. Now, this is the spore stain of Bacillus anthracis. Spore stain could be either Ashby stain or the Schaefer-Fulton stain. The two reagents which are used, or the two dyes which are used, are safranine and malachite green, right? So this is the cytoplasm has taken up the safranine dye and the spores are stained with malachite green. Spores do not take up the gram stain. They appear as empty spaces in a gram stain. So you need this special stain to demonstrate the spores. Spores of Bacillus anthracis, remember, they are only formed in soil and culture, never in the human body. And these are either subterminal or they are central, and they can be round or oval. This is the drumstick morphology of Clostridium tetani, which has terminal circular spores. I hope you can appreciate that the spore is not taking up the color of the purple color. So these are not taking up the gram stain. This is again the drumstick morphology of Clostridium tetani, but this is the spore stain. The spore has taken up the green color, and the cytoplasm has taken up the pink color of safranine. This is the gram stain morphology of Clostridium perfringens, which has subterminal spores. You can see that the spores are appearing as empty spaces. Remember, in pathogenic bacteria, there are only two genera whose members form spores. One is Bacillus, the other one is Clostridium. Of course, Clostridium spores can form in soil, culture, as well as human body. This is the Ziehl-Neelsen stain to demonstrate the acid-fast rods of Mycobacterium tuberculosis. The bacterium has taken up the carbol-Fuchsin stain, and the counter stain is methylene blue. So acid fast rods, meaning they are retaining the primary stain carbol-Fuchsin following decolorization by sulfuric acid. Stain used to demonstrate mycobacteria is the fluorescent dyes auramine O and rhodamine B. This is the auramine stain of Mycobacterium tuberculosis. What is the advantage of using the fluorescent dyes to demonstrate MTB? These are more sensitive than the Ziehl-Neelsen stain, and they take up less time, 1/5 of the time, to detect the fluorescent rods under the fluorescent microscope in comparison to looking for the acid fast rod under the light microscope. Okay, this is the Phyte Farraco stain, which is used to demonstrate the Mycobacterium leprae in tissue sections. So basically the tissue is treated first by the pathologist in a special way, and this is followed by the same reagents as are used for the Ziehl-Neelsen stain, primary stain carbol-Fuchsin, followed by the decolorizer sulfuric acid, followed by the counter stain methylene blue. These are the Actinomycetes, which are classical bacteria which are slender, gram positive, and they show branching like the molds of fungi. Okay, and this is the gram stain which is demonstrating these branching rods. What is this? Can anyone identify which stain this is? The pink colored bacteria and the counter stain, something in the background is blue. So this is again the Ziehl-Neelsen stain, and this is demonstrating the branching rods of nocardia. Okay, under the family Actinomycetes, which are all branching gram-positive rods, we have the Actinomycetes, which are non-acid fast, and we have nocardia, which are partially acid fast, meaning they retain the primary stain of carbylfuscin when we use 0.5 to 1% of sulfuric acid as a decolorizer. So very important difference between nocardia and actinomycetes is that nocardia is partially acid fast. Now this is the CSF sediment of a neonate who is presented with fever, headache, not headache, neck rigidity, and basically neonatal meningitis. Now, which organism are you going to think of in this picture? You can see gram-positive rods in the CSF sediment. So what is this organism most likely going to be? Listeria monocytogenes. Okay. Now, this is again the CSF sediment of a neonatal meningitis case, and we are seeing gram-positive cocci in chains. So which organism is this going to be most likely? This is most likely Streptococcus A galactiae, that is the group B beta hemolytic Streptococcus. These are, of course, gram-negative rods. It could be anything. It could be E. coli, Klebsiella, Pseudomonas. It could be Salmonella. Gram-negative rods. This is the Wesen stain, which is demonstrating the bipolar staining or the safety pin morphology of Yersinia pestis. This bipolar staining is not seen under gram stain. It is only typically seen with the Wesen stain. This image is of the Pund cells which are containing the Donovan bodies. That is the Giemsa stain, which is demonstrating Klebsiella granulomatis, which is the causative agent of donovanosis or granuloma inguinale. So the macrophages are containing the safety pin appearance of the Donovan bodies of Klebsiella granulomatis. So the macrophages are called as the Pund cells, and the Klebsiella, the bacterium, is called as the Donovan bodies. Since Treponema and Leptospira are too slender to be seen by the light microscope, we use the dark ground microscope to demonstrate the spiral-shaped spirochetes. This is Treponema pallidum. This is Leptospira species, which have either one end hooked or both the ends are hooked. Now, since Leptospira and Treponema are too slender to be seen by the light microscope, we can make them thick enough to be seen by the light microscope by impregnating them with silver. So this is Treponema being demonstrated by the silver stain. So either if it is a tissue section, we use the Livardity stain. If it is a film of a fluid specimen like the exudate of the hard chancre, this would be the Fontana silver stain. This is another silver stain. This is a biopsy specimen from this is a gastric biopsy specimen, and the black colored bacteria is the Helicobacter pylori. This is the Warden starry silver stain. Starry silver stain is also used to demonstrate another bacterium, that is Bartonella hensleyi, which is the causative agent of cat scratch disease and bacillary angiomatosis. So whenever we want to demonstrate Bartonella hensleyi in a biopsy specimen, we use Warden starry silver stain, and you can see the black colored bacteria. Now this is a picture of underwater photograph of a school of fish, and the moment we hear school of fish, what are we going to think of? The gram stain morphology or arrangement of Haemophilus ducreyi, the causative agent of chancroid or soft sore. This is demonstrating the flagellae. So can you appreciate the flagella at the single pole. This is Vibrio being demonstrated by the flagellar stain, which is the Leifson and Ryu stain. Flagella are too slender to be seen by the light microscopes. So basically the flagella is impregnated to make it thick enough. And this is called as the Leifson and Ryu stain. Those are the peritrichous flagellae of, it could be E. coli or it could be a salmonella. It could be any bacterium which has peritrichous flagellae. Now, this is the Quellung reaction. And if you see the figure B, this is basically the bacterium which has some refractile structure around it. That's the capsule. This is basically Streptococcus pneumoniae because it's present as a diplococcus, and you can appreciate the capsule. And if we add the specific anticapsular antiserum, the capsule swells up, and that is called as the Quellung reaction or the Neufeld reaction that is shown in the figure A. Basically, the formation of the immune complex leads to the capsular looking swollen under the light microscope. Okay. So with that, we move on to certain biochemical reactions used to identify bacteria. So this is a picture of the catalase test. Basically, enzymes, bacteria have catalase enzyme which has the ability to break down hydrogen peroxide into water and oxygen. And that oxygen is seen as bubbles when we pour hydrogen peroxide onto the colony. Okay. Most bacteria are catalase test positive except the acids. So ASS are catalase test negative. So which is this? A is for all anaerobes. S is for streptococci, that would include the enterococcus, and Shigella dysentery type 1. These are catalase negative. This is a figure of the, or a picture of the oxidase test. The cytochrome C oxidase of the bacteria is identified. Basically, the filter paper is impregnated with a substrate for the enzyme, and development of a purple color on application of the colonies will tell us that this is oxidase test positive. Now, most bacteria are oxidase test positive except CESS. So, C. Corynebacterium, E. all Enterobacteriaceae, and the Staphylococcus and Streptococcus. So these are all oxidase test negative. The easy way to remember is the exceptions. Blindly, all the other bacterium will become oxidase test positive. Whether it be Pseudomonas, it could be Neisseria, it could be Vibrio, it could be Brucella, Bordetella, etc. They will all be oxidase test positive. This is the Hugh Leifson's oxidative fermentative medium, which tells us whether a bacterium breaks down sugar, glucose, and if it does break down sugar, whether it is breaking it down under aerobic conditions only or both aerobically as well as anaerobically. Okay. So anaerobic conditions are provided in the left test tube, as you can see, over which sterile petroleum jelly has been poured. And after overnight incubation, if we see that yellow color, that is acid, has been produced in both the test tube, that tells us this is fermentative utilization of glucose. If acid is developing only in the aerobic test tube, that means it is oxidative utilization of sugars. And if there is no yellow color, no acid produced in either of these test tubes, that means this bacterium does not break down sugars. So some bacteria like Campylobacter, Helicobacter are unable to break down sugars. They will show the figure 3 of this. Okay. So figure 1 is fermentative utilization, figure 2 is oxidative utilization, and figure 3 is no utilization of glucose. That's an image of the urease test. Urea gets broken down into ammonia and carbon dioxide. Ammonia produces alkaline conditions, as a result of which the pH indicator undergoes a color change. So there are some bacteria which produce the urease enzyme. The most important ones to remember is Helicobacter pylori, Proteus, Urea plasma, and Cryptococcus neoformans. Of course, that's a fungus. IMVIC reactions are used for the identification of gram-negative rods, especially members of Enterobacteriaceae, indole test, methyl red test, Voges-Prosker test, and the citrate utilization test. Briefly, the principle of each of these is indole test is ability to convert tryptophan to indole, basically production of the tryptophanase enzyme. Methyl red test is that when this bacterium utilizes sugars, it produces so much amount of acid, the pH remains less than 4 for a very, very long time. So mixed acid pathway is used to break down these sugars. In the Voges-Prosker test, yes, this bacterium breaks down sugars, but it does not utilize the mixed acid pathway. So it produces acetone. So acetone production is detected by the Voges-Prosker test. And last is ability to utilize citrates as the sole source of carbon. So IMVIC for say a bacterium like Escherichia coli would be plus plus minus minus. For a bacterium like Klebsiella, it would be minus minus plus plus. That's an image of the coagulase test. To be more specific, the tube coagulase test, which detects the free coagulase enzyme. You can see the positive result in the upper test tube. So it basically plasma is taken and a few colonies of the bacterium is added. And after 4 hours of incubation, we look for coagulum formation. Okay. So basically coagulase test would be positive for Staphylococcus aureus. So all staphylococci are divided into two groups, coagulase positive, that is Staph aureus, and the rest are called as coagulase negative. That's our slide coagulase test, which is used for detection of the bound coagulase of Staphylococcus aureus. This is the motility test medium. So it's a semi-solid medium, which is stab inoculated with the colonies of the bacterium, and after overnight incubation we look for the appearance of this kind of translucency which has spread along the spread towards away from the line of inoculation. That will tell us it is positive, meaning motile. And if we just see that the bacterium has formed colonies just along the stab inoculate, we will say it is a non-motile bacterium. That's the figure that I have drawn to explain this further. The left one is a motile bacterium, the right one is a non-motile bacterium. This is the CAMP test, Christie Atkins Munch Peterson test. Basically, a blood agar plate is taken and we take a few colonies of Staphylococcus aureus and streak them vertically. And then at right angles, we are going to streak our Streptococcus A galactiae. And after overnight incubation, the hemolysin, that is beta hemolysin of Staphylococcus aureus, synergizes with the hemolysin of Streptococcus A galactiae to produce arrowhead hemolysis, that is enhanced zones of hemolysis. The same CAMP test is also positive for Listeria. So listeriolysine, the hemolysin of Listeria, synergizes with the beta hemolysin of Staphylococcus aureus to produce these similar zones of enhanced hemolysis. Coming to the reverse CAMP test, here what is reverse is that it is Streptococcus A galactiae which is streaked vertically first instead of Staphylococcus aureus, and at right angles we are streaking the colonies of Clostridium perfringens and enhanced zones of hemolysis are produced. Okay, this is the PYR test. PYR is basically detecting the enzyme pyrrolidonyl amidase test enzyme. The in the PYR test, remember, um, it is positive for Streptococcus pyogenes and Enterococcus. Okay, so Streptococcus pyogenes, that is the group A beta hemolytic Streptococcus, is positive for PYR and Enterococcal species. Okay, coming to this figure, this is the bile esculin test. It is testing two important characters of the bacterium: ability to grow in 40% of bile and ability to hydrolyze esculin. So, right, we can see this kind of brown color appearing on this white colored medium. Bile esculin test is positive for Enterococcus and non-enterococcal group D streptococci like Streptococcus gallolyticus. So very important for Enterococcus and Streptococcus gallolyticus. This is the string test of Vibrio. Basically, we prepare a suspension of Vibrio on a slide, and with an inoculating loop, just take a drop of bile salt and mix it in that suspension and take out the loop. It comes out with a string attached to it. String test in microbiology, in bacteriology, that is positive for hypermucoviscous strains of Klebsiella pneumoniae. They produce so much amount of capsular material that if you just touch the colonies with an inoculating loop, and as you remove it, it comes out with a string attached. Basically, that's the capsular material. This is the dilution method, broth dilution method of antibiotic sensitivity testing. So here we have the ELISA plate and we have the wells of the ELISA plate containing uh, increasing concentration of the antibiotics, and we add a standard inoculum of the test bacterium, and we determining which is the lowest concentration of the antibiotic, which just inhibits the growth of bacterium. So that will be called as the minimum inhibitory concentration. Is the agar dilution method of antibiotic sensitivity testing. I have just shown one Petri plate. Actually, there are multiple Petri plates, each of them containing a different concentration of the antibiotic. And at specified sites, we are going to apply the colonies of the of different test isolates and determine the minimum inhibitory concentration for all those isolates, right? So dilution method exactly can quantify the with the minimum inhibitory concentration, agar dilution or broth dilution. But these are pretty tedious to do, time consuming. Not everybody has so much time to carry out this method. So a easy to do method of antibiotic sensitivity is the Kirby Bauer disc diffusion method, where we have antibiotic discs which are impregnated with a single standardized concentration of the antibiotic, and we measure the zone of inhibition around the antibiotic disc and refer to standardized tables to tell us whether it is resistant, intermediate sensitive, or sensitive to that particular antibiotic, right? So on each plate, these are not just one single disc of one antibiotic, these are multiple antibiotic discs. This is an image of the Stokes disc diffusion method of antibiotic sensitivity. Here, the test strain and the control strain are swab inoculated on the same Petri plate, and instead of referring to standardized tables, here the zone of inhibition around the antibiotic discs of the test strain is compared with that of the control strain to say it is resistant or sensitive to the antibiotic or not. Of the disc diffusion and the dilution method is the epsilometer test or the E-test. Here, instead of discs with single standardized concentration of the antibiotic, we have plastic strips with graded concentration of the antibiotic, and where the zone of inhibition intersects the plastic strip, that reading is taken as the minimum inhibitory concentration of the antibiotic. Now, this is a disc of optogen, and you can see a wide zone of inhibition around the optogen disc. This is the optogen sensitivity testing. And of course, if the zone of inhibition is more than or equal to 14 millimeter, we will say our bacterium is sensitive to optogen. And the moment we think of optogen sensitivity, which organism should strike us immediately? Streptococcus pneumoniae. This is a commonly used test to differentiate Streptococcus pneumoniae from the viridans group of Streptococci because both of them are alpha hemolytic on blood agar. This figure shows both optogen sensitivity and bile solubility testing being done on a blood agar plate. You can see the wide zone of inhibition around the optogen disc, and you can see the bile solubility test being done where the arrow has been put. So here what we are doing is over the colonies, over a few selected colonies, we are just going to pour a drop of bile salt, and within a few minutes we will see that the colonies are just disappearing because addition of bile salts has increased the activity of the autolytic enzymes. Again, a test used to differentiate Streptococcus pneumoniae from the viridans group of Streptococci. Streptococcus pneumoniae is bile soluble, whereas viridans group are bile insoluble. This figure shows the bacitracin sensitivity testing. You can see the zone of inhibition. Any zone of inhibition around the bacitracin disc, we say it is sensitive to bacitracin. This is positive for Streptococcus pyogenes. This is also called as the Maxted's observation. This figure shows us novobiosine resistance. Okay, this is a test used to identify two important species of Staphylococcus, that is Staphylococcus saprophyticus and Staphylococcus xylosus, which are resistant to novobiosine. So if we see that the zone of inhibition is less than 12 millimeter, or there is growth right up to the edge of the disc, we say it is novobiosine resistant. All other Staphylococcus species are sensitive to novobiosine. This figure shows phage typing. I'm not going to explain the whole phage typing procedure. Basically, the bacterium is swab inoculated over the Petri plate and over the culture medium, and a specified amount of phages are placed over the plate, and after overnight incubation we will see these plaques of inhibition. Okay, and depending upon the spectrum of the phages to which the bacterium is sensitive, meaning it has been lysed by the phages, we will type the bacterium. Okay, typing is basically intraspecies strain characterization. So phage typing classically has been done for Staphylococcus aureus, Vibrio cholerae, Salmonellas, Mycobacterium tuberculosis. This figure shows the VDRL slide, which is a slide flocculation, which is done for the diagnosis of syphilis, one of the non-treponemal tests, slide flocculation test. So basically, for this, there are 12 wells in which we are going to prepare serial dilutions of the patient's serum and the cardiolipin antigen and look for flocculation under the light microscope. That's an ELISA plate. We are well familiar with it. ELISA is used for detection of antigens in specimens, antibodies in sera specimens. An ELISA plate we have earlier used in the dilution method, the broth dilution method. It's used also for nation tests. Some viruses have the ability to agglutinate certain RBCs like sheep, horse, or guinea pig RBCs. And if so, basically, heme agglutination tests can be used for detection of viruses in specimens or even antibodies in sera specimen. So if you see the button formation, it means it's a negative heme agglutination. If the button does not form, it means heme agglutination is positive. Okay. This figure shows the immunochromatographic tests, or also called as the lateral flow assays, which are very rapid tests, which can be used for detection of both antigens in specimens or antibodies in serum specimens, often used for diagnosis of COVID-19, malaria, as well as leishmania. So the malaria rapid diagnostic test that you may have heard of, these are all immunochromatographic tests. If you ever see a machine like this and you read something like a thermocycler written on it and you're asked the purpose of this, think of polymerase chain reaction, which is done in a thermocycler. Of course, you must be remembering the steps of polymerase chain reaction: denaturation, followed by annealing, followed by extension. That's a biosafety cabinet or the biosafety hood. That's the worktop or the workplace where the microbiologist works. It has a laminar flow of air to prevent infection being acquired by the microbiologist and to prevent the spread of infection to the community, right? So that was some important images of bacteriology. I will continue with some more videos on more images. I will leave you today at this time. Small uh, advice to all of you that we are all passing through tough times when we are preparing for our PG entrance exams. But if you believe in yourself, you can achieve anything. If you believe that you can do it, 50% of your job is done, and the rest of the 50% is going to be done by discipline and hard work. So self-belief, discipline, and hard work, anything is possible in this world. Bye-bye.